Okay, so I guess it's up to me to announce, and it's a great pleasure to be with you guys, at least mentally in Merida or Guanajuato, or anywhere else in Mexico. Uh, Juan Carlos Diaz. So we met multiple times in various occasions, sometimes in Mexico, sometimes in other countries. Um, Juan Carlos is topologist by training, but he's involved in this line of research, joining topology and neuroscience, and I think that's a very important line of research. So, human brain connectome and how to understand it on different levels using topological data analysis. This is what we are going to hear today. Juan Carlos, my great pleasure. Floor is yours.
Thank you, Pablo. So I am going to talk a little bit about the things that I have been working in the last years. These are the applications of TDA to the study of the human brain connectome. And this is a joint work with Dr. Zeus Gracia Tabuenca, Isaac Arelio, and Mozzarella Cauter. So this is the original team. We started as a group of friends that we share our meals every day. And at some point, I asked to Sarah if he was interested into this kind of applications of TDA to neurosciences. And he answered that he was interested, but he was more interested not just learning, but publishing a paper. So, well, this is our teamwork. These are very old pictures. All the people involved here now have a lot of changes, but it's okay. Well, I'm going to start speaking a bit about MRI, magnetic resonance imaging, and this is related to the BOLT signal. BOLT is an acronym for blood oxygen level dependent, and it's related to neural activity. So I'm going to speak a little bit about this. Maybe you have some regions of the brain that you have a set of neurons that are asking for a supply of blood with oxygen. So the brain responds. This is a physiological process and sends blood to this region of the brain. And usually it overcompensates the amount of blood that is needed in this region of the brain. And this is a phenomenon that is detected in the MRI machines. The magnetic machine that is used to measure the magnetic field is very sensitive to these kind of changes. And this is why it's important in the study of neurosciences. And also, the brain is divided into a mesh, a cubical mesh, and where each cube is called a voxel, just like a reference to the word pixels that is used to draw images in 2 dimensions. So the brain is divided into voxels, and the volt signal is measured in each voxel over time. There are several ways to perform a study. For example, the task-related fMRI. And in this kind of studies, you ask the participant to do an activity for a short period of time, maybe just moving your hand. So this is depicted here. So you ask the participant move your hands over a period of time, maybe 5 minutes. Then just rest, don't do anything in that period of time. Then move again your hand and repeat this maybe like five times. And there is an expected blood signal that we are trying to measure in each region of the brain. So the expected blood signal is just like a peak of this curve drawn here. And it's going to be a reference of the activity of the brain in some regions. So it's very useful to study brain functions. And what is studied is this kind of images. In the background, we have a structural image. It's a 3D image of the brain. but with higher resolution. And the things that are colored here are a statistical map of which voxels have concordance with the volt signal. So we have, for higher concordance, we have the color of white. For lower concordance, we have the red color. And with no concordance, nothing is wrong in the rest of the brain image. So this is pointing us that this region of the brain is related with this activity that we are studying. And well, this is the kind of studies that are done, task related. And I wanted to show you this image that is the software that is commonly used to study fMRI images. In the background, we have again a picture of the brain, but this is not a picture of a real brain. It's just a visualization, just to identify regions. And again, the colored image is depicting the statistical map that is pointing on regions of the brain that are in accordance to the task activity. So there are another kind of studies that is the main purpose of our studies that is called resting state fMRI. And this is used to measure spontaneous activity in the pulse signal. And the instructions for the participant is just don't fall asleep, don't close your eyes, and try to remain still during the scan process. The processes are around 6 to 10 minutes. And the instructions are very important because if you close your eyes, the functional activity in the brain changes and is related to the process of sleeping. So, and the second part that remain still during the scan is also important. If you are closing your eyes and you are sleeping, it's easier to fall asleep inside the machine because the machine is always giving a constant sound that at the beginning it's annoying, but if you're staying there for more than 5 minutes, your brain just comes to ignore the noise in the machine. And it's easy to sleep if you're closing your eyes. And some people snore when they are sleeping. So this this is the reason why you need to remain still during the scan because this is going to affect the study. So these are the instructions for resting state, and well, we are going to see a little bit of these studies. So in general, we have these two kind of images, structural MRI that have a high resolution. We have a very detailed image, 3D image of the brain, but it's static. It's just a 3D image of the brain. And it's commonly used to detect lesions of the brain. So you have also you can identify white matter and gray matter in the brain, and you can, if you are a trained physician, you can identify regions of the brain. And the functional magnetic resonance images that have lower resolution, lower spatial resolution, but they are taken over a period of time. So it's more like a video of the brain, and you can detect differences in some regions of the brain that are working in concordance. So these are the kinds of studies that you can do with these machines. And also, if you want to study the brain, it's very useful to use a parcellation of the brain. There are a lot of ways to parcellate the brain according to what are you trying to study. So we're going to focus only on these kind of parcellations that are related to anatomical regions, to homogeneity. This means that voxels that have the same functional behavior over a period of time are colored with the same color. Functional, it's more or less the same as homogeneity, but you are only focusing on specific regions of the brain. So it's not a parcellation of the whole brain, but it's you are choosing specific regions of interest that can that can reveal the all the structure of the uh, brain connectome. So this is a brief introduction of the MRI studies, and now I'm going to talk a little bit about the mapper algorithm that, well, I think a few of the participants in the meeting are very familiar with this algorithm. So in this case, you have a set of points. This is an illustration in this paper. So you have a set of points that are drawing the surface of a hand. So the next step is using a filtering function that is used to color the hand in this next image. So traditionally, well, this is an example. You are just projecting the dots into the x-axis. So you have a lot of dots here. And the starting point is here in 0. So the color red is associated with colors, the color red. And if you're moving to higher values on the x-axis, you're just changing the values of the colors of the hand. So, for example, this point here corresponds to color orange, and etc. And the next step is to give a binding that is just a set of intervals in the x-axis. I'm going to draw it with another color. So you are maybe choosing this interval, maybe another interval, and it's required that the intervals are overlapping. There are several ways to construct this, but now you are going to focus on the pre-image of each of these intervals. And this is what is drawn here. The pre-image of the first interval is the tips of these three fingers. And the next pre-image is this drawing of the fingers, and etc. And the next step is choosing a clustering algorithm. Each of these points, well, maybe they are in the same pre-image of two intervals. This is done with this intention, because this is important, that they have some kind of overlapping. So the first tip is going to be clusters with some kind of algorithm. And this cluster is represented here with this red dot. Another tip of the finger is another cluster. So it's drawn here with another dot, red dot. And we have three dots representing the tips of the first three fingers. In the second step, you're going to build another clusters for the next pre-image. And you're going to do this in all the pre-image. So you have these clusters represented here. And you're going to join each of these clusters if the pre-image have dots in common. So this, I'm going to draw it. Maybe this dot is the same as in this region, and the clusters are going to be connected in the mapper graph. So the idea is that you're building a graph, a combinatorial object that represents the topology of the original surface or set of points over a cloud point. So this is an example. So this tree is representing the same topology as the hand, just as an example. So we are going to see how the mapper algorithm is used to study magnetic resonance images. This is a paper of Manish Sagar and this long list of collaborators, in which you may identify Gunnar Carlson, that is one of the principal researchers in this field. And this article was published in Nature in 2018. and it's an application of the TDA, the mapper algorithm, to study magnetic resonance images. So in this study, they studied 18 subjects. They have a task related study that was run for around 24 minutes. And it's a continuous multitask paradigm. They asked the participants to rest a little bit inside the machine, then do a math work that is only subtracting or adding numbers, small numbers between 0 and 10. Maybe this is interesting for mathematicians. And then they are asked to answer the, the problem, the arithmetic problem. And the machine have a device that is used to record if the answer is correct or is it wrong, and also records the time of response. So you can measure these two things. A working memory activity that is they are showing you a set of figures, a sequence of figures, and the activity is that you are needed to remember the figure that is two images behind. So if, for example, they are showing you a star, you need to remember if the figure, the previous two figures was a star. They are also recording if you're having right and wrong answers, and the time of that you take to answer the question. And also video. The video activity is they are showing you a video with, in this case, it's a fish tank. Just there are a lot of fishes there, and they are moving. And at some point, a signal appears in a determined kind of fish, and you're asked to press a button if you see the signal in that kind of fish. And another signal if there's appears in another kind of fish. So they are recording also the response time and the accuracy of the answers. So this is the general study, and we are going to see how they use the mapper algorithm. So you have in this case, you have a complete image of the brain every 1.5 seconds. So in total, you are going to have 1017 images of the brain. And this is the how they look if you are taking these studies. For each, then the next step is just organize the information in a way that you are going uh, uh, to work with this high dimensional data set, and you want to utilize the mapper algorithm in some way. So in this case, they have arranged all the voxels of each of the images in a row, like in a vector. So they are just arranged here. You're forgetting the spatial ubication. Just put all the voxels in a row. And this is for the frame number one. For frame number two, you're arranging again the set of voxels, and you have a very high dimensional space. And the next step is using a dimensional reduction algorithm in order to reduce this number of dimension, 271 hundredths, and you're going to reduce this dimension to only two. So like in this drawing here. So in this drawing, you have again 117 dots. Each one represents one frame of the study. And the reduction algorithm is, in this case, they use some variation of stochastical neighborhood embedding, it, because they wanted to preserve local properties of the network. So they want that two similar images of the brain remain closer in the study. So, well, this is the next step, the dimension reduction. The next step is going to use the clustering algorithm. So you need to divide the, the two-dimensional space in bindings, and each binding has an overlapping, and you are running the reduction algorithm in each of the binding. That it's, in this case, there are cells, like square cells. The clustering algorithm it's depicted here. So these three dots are in the same cluster according to your algorithm. And each of the points represents one of the images that the study has done. And then you build the mapper graph, as we have seen in the previous slide. So these slides are, the dots are connected. Well, I'm going to say this cluster and this cluster are connected if they share a point in the original dataset. So you have now the mapper graph for each of the participants, and now we're going to see how they study these mapper graphs. So this is the result for one of the participants. The filtration that I used in this case is the time frames from 1 to 1017. So the first points are colored in blue, just the instructions that are given to the participant. The instructions are given previously, but it's always useful to repeat the instructions in a very short presentation, maybe just 1 minute, just to remember the participant what are the activities that they are going to do. Then in color green, there is the resting state phase. Maybe for 5 minutes, they are asking the participant just stay awake for this period of time. And then they are asking to do the tasks. First is the working memory in color yellow, the video in color orange, and the math in red color. So this is the mapper graph, and we can see here that all the tasks that are related to answering some questions are clustered in this big part of the graph. And the resting state are in these kind of branches of the graph. And also the working memory, some of the clusters are outside the big cluster that is drawn here. So this graph is showing us the dynamical behavior of the study for this individual. So as this is a graph, now you can measure some graph properties, in this case modularity. These are the images for two of the participants, subject 14 and subject 07. The modularity values are shown here, 0.38 and 0.57. These values suggest that there are higher modularity in the mapper graph. And they wanted to compare the modularity versus the correctness of the tasks that they answer the tasks. So we can see here that they have a positive correlation. These are the results of the adjustment. And well, for example, the subject that has here a modularity around 0.6 is this case, the subject 7. And the percentage of accuracy is around 94%. So this is the point that is drawn here. And also they tested for the response time. This is a negative correlation. And the same subject seems like this subject was taking a little bit more answering the questions, and it's reflected in the accuracy of the answers. So maybe this participant was really focused on trying to do the activity very well. And well, this information is we can see this behavior in these graphs. On the percentage of missed answers is wrong here, but I think this it's natural that this is the opposite as the percentage of corrected answers. So this is the kind of information that you can extract from the mapper graph. In this case, they wanted to study the dynamically behavior of these studies, how they changes over time. Well, now I'm going to talk a little bit about persistent homology, the persistent homology approach, but I'm going to focus only on the pericurves. In this case, we are building a network, but not like in the previous case that each point, in the previous case, each point represents a frame that contains all the information of the brain. Now we are going to focus on the whole time series. So for example, this is some kind of parcellation of the brain. I am going to focus only on determinate regions that are these dots. I have a time series for each of these dots that is shown here. And I'm going to compare these two time series using Pearson correlation. There are more ways to compare this, but this is like the most common approach, just using Pearson correlation. So I'm going to build a network, and each of the regions of interest is going to be a node in this network. And I'm going to draw the links on the network as a weighted link, and I'm going to use the correlation between these two regions of the brain. So this is a weighted network, but the values goes from -1 to 1. So in order to use another algorithm, just to study to compare these networks, we transform this information using this 1 minus r. 1 minus r is just 1 minus the correlation of each of the links. And this transform the values that goes from -1 to 1 into values that goes from 0 to 2. And the positive correlation values are going to be closer to 0 with this transformation. This transformation does not satisfy the triangle inequality, so it's not a metric in the mathematical sense, but it still is useful to do all the computations on persistent homology and other things. And the next step is just computing the pericurves. The pericurial curve counts the behavior of the connected components of the network. It starts in this case with 264 set of points, all disconnected. And over a period over a short period of the filtration values, the network is beginning to connect, and these are the steps that are shown here in the very zero curve. The very one curve is counting the number of holes of dimension 1, and is drawn here in color red. And the very two curve is counting the holes in dimension 2. So this is well, this is the behavior of these very zero curves. If you want to compute the higher dimensional Berry curves, they are going to be translated a little bit more. Well, this is an interesting behavior, and some people have studied this using the Euler curves instead of the Berry curves. Well, I wanted to give you the drawing of the minimum spanning tree because the minimum spanning tree is closely related to the Berry zero curve. So each of the steps of the Berry zero curve is one of the links that is drawn in the minimum spanning tree. Well, I am going to talk a little bit about this later in this talk. So we wanted to use this model, this approach. I'm going to give you an example of 100 sets of 100 points. This is only a set of 100 points. We repeated this with 100 times. And these are uniformly distributed in a square. And this is the square that is drawn here. And we're going to do more or less the same, but instead of they are uniformly distributed in a cube, the radii is going to be normally distributed. So around the number zero, we are changing the distance of the points, but using a normally distributed proportion. So these two datasets can be analyzed using the Berry curves. For the uniform Berry curve, it's the darker green. We can see that it's considerably different as the normal distribution of the radii. And also for the Berry 1 and Berry 2 curves, we can see differences here. But we want to do this like in a statistical way. So we want to do a statistical test just to compare these two populations. And we can see that there are statistical differences just because, well, this is a toy example. But we have found differences in dimension 0, dimension 1, and dimension 2 when we were computing the area under the curve. So in our first paper, we wanted to compare these curves, and we weren't really sure how to compare these curves. So one option was the area under the curve, that is the L1 norm, and it's useful to compare functions in general. We also used the kurtosis, that is a measure of this, how is this similar to a normal Gaussian curve. And well, we use the kurtosis and also the slope. The slope for this, we use it like if this was a set of points, and just compute the adjustment, the line that goes for better for this set of points, and computed the slope of this line. And well, these are different ways to compare these curves. And we used this approach to study this set of fMRI images. So in this case, we have a set of children diagnosed with ADHD, which is attention deficit hyperactivity disorder. They have a study of MRI and also a study with a psychologist, and that is he's giving a test for the children, and then the specialist is able to determine if the children has ADHD or not. It's very it's one of the main problems studied in neurosciences, just to give a way to detect these diseases. So this is cataloged as a developmental disorder, and is related to the difficulty to control their own behavior. The main of the studies that are done in this field are done with graph theory. So this was before the introduction of TDA techniques. And the regions of the brain that are related to this disorder are the frontoparietal attention systems and the default mode network. And we wanted to compare the very curves of these two groups. We have a group with typically developed um, children. We computed all the very curves, the very zero curves. And we have another group with children diagnosed with ADHD. And also we computed all the very curves. And we found that using logistic regression, that the area under the curve of the ADHD group is lower than the typically developing children. We tried this with 4 different atlases, and we have the same results. So our result is robust. And also we compared with a null distribution that just is a way to say that our information is related to a biological system, and it's not a random uh, set of information. No? So these are the comparisons of the area under the curve, the kurtosis, and the slope. But we didn't find differences in the kurtosis. This means that in general, all the zero curves are very similar, and also they have also the same slope. But the area under the curve, this study shows differences between the two groups. And also we did this for specific regions of the brain. So this was the global comparison. This was a comparison just in determinate networks. The first one is just using the anatomical parcellation of the brain, and the squares that are drawn here represents that there are statistical differences on the log odds ratio, and it's always in the same direction. The area under the curve of the ADHD is lower than the typically developing children's. And the networks that are studied is parietal and frontal. This is the line that is drawn here. The cerebral and the frontal. Well, all the regions that are linked with the frontal region. That is in concordance with the previous studies. And also for a different kind of parcellation, that is a functional network, the previous studies that shown that the regions of the brain that are more related to this disorder are linked with the default mode network that is shown here. And also, we have found the same differences between these study groups. And these are the networks that are related to the default default mode network. Well, that was our first work. Then we wanted to switch to another study. That is how the functional connectivity is changing over time. And this was done here in Querétaro with a sample comprised with 98 typically developing participants, 53 females, 45 males, over the range of 6.7 years to 18 years. Of those, 41 returned for a second session and 16 for a third. And this was obtained here in Querétaro. And this is, well, we wanted just to study how are the changes over puberty in these children's. So this is just an image that is shown. This is showing how the data set are obtained. The lines corresponds to the same subject. For example, this subject was tested 3 times. One was around 7, maybe near to 8 years. then when he was maybe 13 years, and the last time when he was 16 years. Blue lines, well, turquoise lines corresponds to the male participants. The pink color is related to female participants, and QC is quality control. So these uh, dark points just they were excluded, analyzed in a posterior study, but these are not included in the original sample. Well, the line that is drawn here is correspond to the mixed linear model, and the the scales here corresponds to the age of the participant and the pubertal development status. So this is like a clinical diagnosis. Well, if the participant doesn't have any signs of puberty, it corresponds to the level 1. If have some signs of puberty, then corresponds to level 2, and etc. Until the last step, that is a person that has already passed through all the puberty changes in the number 4. So this is just a test. They ask the participant, just answer these questions, and this is how they compute the development status. So this is a curve that is representing all the changes in the population here. We tried to compare the very zero and the very one curves. In the first image, in the x-axis, we have the pubertal development status, just 4 steps. And we computed here the very zero, the very zero area under the curve. Again, we analyzed this with a linear mixed model, and we have seen here that this peak in the curve suggests that there are a lot of changes in the connectivity of the children's between the pubertal development status from 2 to T, to number 2 to number 3. And well, this is it's a good description of the changes in the brain. But if we use the age as an indicator of the changes in the functional connectivity, it's not as clear as the pubertal development status. And this is because some kids enter to puberty at different ages. So the pubertal development status is a better descriptor for the changes in the puberty of each of the children's. And we already seen here also the changes using the very one curve, the area under the curve, and these are shown here. So the nonlinear trajectories shows us the lower area under the curve at the end of the adolescence and higher connectivity in relevant ages. So the relevant ages are between 2 and 3. It's a better description based on the puberty stage. And what the inflection point is at the puberty onset. Well, this is we published this a few years ago, and now I'm going to talk a little bit about these research questions. This comes because we gave a talk in a seminar, and one of the participants was from the area of computation. I'm not really sure what kind of specialization was this researcher, but he asked, well, the fMRI datasets looks like really noisy datasets. So it looks like you have only noise and you're just analyzing noise between all these studies. And well, then they come with these questions. How reliable is the information obtained from fMRI? Do they change a lot between sessions? Maybe from one day you have some kind of information and the next day you have a completely different kind of information. What is the variability of the topological and graph features of functional networks? And lastly, is there a network feature that can characterize individuals, the individual's functional brain connectomes? And we try to answer these questions. And for this, we used the Midnight Scanning Club. It's a data set with 10 preprocessed resting state fMRI studies from 10 healthy adult subjects. These are 100 studies in total, 30 minutes each study, 5 males and 5 females between ages from 25 to 34 years. And it's also reported in this paper that subjects 8, 9, and 10 have closed their eyes during sessions and they have extreme movement. So subject number 8 was probably snoring during sessions. This is just like a folklore about this data set. I'm not. This is not. This information is not included in the paper, but well, this is a very well studied data set. And the name of the data set, Midnight Scanning Club, is because the studies were performed at midnight because it's cheaper to use the magnetic resonance image scanner at this time of the day. And probably they were the same people of the laboratory, maybe graduate students, doctoral students, PhDs, and etc. This corresponds to the ages of the study. Well, again, we computed the Berry curves for each of these participants. We have 10 Berry curves for each participant. And we can see that the variability between Berry zero curves are relatively lower. For this subject, we have almost, well, it's a better example. This is subject number 5. We have almost the same Berry curves for each study. I forgot to say this. In this case, they tested in consecutive days. So they asked the participant come at the laboratory at midnight. We're going to do a resting state study for 30 minutes. And then the next day, they're asked to do the same thing, etc. So each of these lines corresponds to each of the days. But we can see here, just by visual inspection, that subject number 8 have a lot of variability between sessions, and also for subjects 9 and 10. So maybe the subject number 10 was more tired in this day and have different results for the study. So we compare the area of the curve of the very curves for dimension 0, dimension 1, and dimension 2. And we can see that there are differences between subjects and a lot of variability again for the subject number 8. And well, this is like doing the same analysis as we done before, just computing the very zero curve and computing the area under the curve. But we wanted to do something different and something that is clearly identifying the differences between subjects, because maybe in this case, subject 7 have very similar information as subject number 10. And we wanted to have information that separates these subjects. Okay, this is the same picture. And this is why we wanted to use the minimum spanning tree. If we analyze closely the very zero curve, this detects changes on the pituitary ribs filtration only when a connected component is joined with another connected component. At the beginning, you have 10 nodes, and in the first step of this function, you are connecting these two nodes, but this filtration value corresponds to the weight of this edge. So essentially, you are executing one of the algorithms used to compute the minimum spanning tree. That is the tree that connects all the nodes in the network with the lowest weight. And we then we came with this idea to vectorize the information of the network using the minimum spanning tree. And then we compared, there are two ways to study the minimum spanning tree. One is binary, just saying, well, we have one edge here, just put weight number one to this to each of the edges, and the other is remain the weight that was original set in this edge. So we compare the distances between these networks using the Manhattan distance for the binary case and the Euclidean distance for the weighted minimum spanning tree. And this picture here is showing us the differences, well, that information between the same subjects are very similar, but comparing this information between participants is different in general. So we compare the L1 distances. I'm almost going to finish my talk right now. We found that the the variability within the same subject, within subjects is this is lower than the variability between subjects that are drawn here, the means of each of the comparisons. So this is the Cohen's d value, and in this case we have a lot of differences between the comparisons of the minimum spanning trees. And the next step, just using an algorithm to reduce the dimension of these large vectors into a two-dimensional data set. In this case, we use the UMAP projection algorithm, and this is done for the binary case. And in this case, we see here the separation that is a good separation of the information of the subjects. So it's very well identified that each of the dots corresponds to one of the subjects. And for the weighted network, we have the same results, but maybe it's not as clear as the previous one. And the mixed, we try to analyze this. That is just the concatenation of the two vectors, of the binary vector and the weighted vector. And we have three images. This was a suggestion of Pavel because graphs, and I wanted to explore the projection of these high-dimensional datasets using the cluster graphs. So in a high-dimensional space, you have a cluster. In this case, the clusters are very well represented for each individual. And you're going to leave only the edges that is not redundant in the information of the network. And this is how they look. I need to make bigger this image. And again, the subjects that are related to maybe they were sleeping or something like that, they're separated. Subject number, this is subject number 8 is separated from the rest of the participants. And we have the same behavior here. And well, I'm going to finish here. The area under the curve of the Betty curves showed lower variability within subjects and higher variability within subjects, with exceptions of subjects 8, 9, and 10. It's a robust technique to study brain topologies and typical development. Okay, the minimum spanning tree is a more specific way to differentiate individuals' functional connectome, and the topological and graph features are relevant, especially the ones that are interconnected, like the minimum spanning tree is related to the very zero curve. That's all. These are the references, and thank you for your attention.
Thank you very much. So, any questions to Juan Carlos?
Yeah, I had a question in that ADHD study. You used the word brain atlas. So it's a brain, you had two different brain atlases. Are these brain atlases kind of standard brains to which you can then compare subject brains? What is a brain atlas?
Okay, brain atlas is a parcellation of the brain. So we wanted to compare three kinds of parcellation of the brain in order to say that our results are robust, that are independent of the choice of your atlas. Okay, there's a delay here with the information. So we use the anatomical atlas and then another Craddock atlas. This has several ways to be defined. One is with 200 points, one is with 400 points, etc. So we wanted to compare our results, computing the very zero curves for these different atlases, and just to say that it's independent of the choice of your atlas.
So the brain atlas essentially puts a coordinate system on the subject brains so you can tell where they are in relation to each other?
Yes, because we want to compare the networks between subjects, and this is a standard way to say, well, you have like a helmet. This is the same helmet for all the participants. There are a little bit of differences between subjects, but this is a way to compare the brain networks.
Does it involve choosing a diffeomorphism between the subject brain and the atlas itself?
Oh, I'm not really sure. Paul is.
They used to call it brain warping. We'd warp, you'd have a standard brain, and then you take a subject brain and build a diffeomorphism from the
Yes.
brain to the standard one. Do you have to do that in these studies?
Yes, this is the preprocessing part of the study, because all the brains are different, and then you have to arrange all in a similar way just to fit this each brain into a common brain in order to compare all the differences. So yes, you have to have a diffeomorphism.
Yeah. Thank you.
Thank you.
So just to follow up, one of the reasons is it's really a denoising procedure. So you have orders of millions of voxels, but then you group some voxels together, they are geometrically close and in the image of diffeomorphism of a region of an ideal brain. And then, the signal is kind of averaged. That kills a little bit of the noise we have. Just to follow up on the questions. Any further questions, please? I may have two questions or maybe two suggestions. I'd be happy to have a follow-up call at some point. So in the first part, you used Betty curves essentially to show that there is a populational difference between subjects, ADHD children versus test children or a control group.
Yes.
So the important thing I would suggest you to also follow up on is it's important in this type of psychological tasks to have a quantitative way of measure certain things, which are typically measured behaviorally. So you see that child doesn't keep an eye contact. That's probably autism spectrum disorder, and certain things may be an indicators of ADHD. Over here, you have an indication that those groups are different, but now here comes the classification questions. So have a new children in the group, and you make the same measurements. And the question is which group the children belongs to. So one suggestion will go this way. We did some work in topological hypothesis testing, so I'll be happy to send you the paper later on. That's a comment or a suggestion to the part number one. The comment and the suggestion to the part number two would be, you kind of did the mapper plots subject by subject. Well, what if you actually embed all the subjects of the information into a point cloud and then use the coloring function to say, okay, that's the subject number one, that's the subject number two, and see how they are relative to each other. I think that also may be something interesting to see how which regions of all the brain's activity that are being measured are occupied by each individual subject.
Yeah, I have already tried the mapper algorithm with another data set, but it's very similar to the ADHD data set. And the results that we found there are no differences in the mapper graph. So it's not like one set of the control group is in one position of the mapper graph and the study group is in another part of the graph. It's all melted. They're all mixed. So I think that this is because they are very similar. Essentially, they are very similar. So we need to find another approach to find the differences between each of the brains.
The differences are subtle. That's the key point.
Yes.
And from my experience with EEG data, there is also a problem of generalization. So you train your model on data coming from one fMRI machine, and then go to another city, to another fMRI machine, and try to use the same algorithm. And I guarantee it won't be as efficient as it used to be for the first one. That's an exercise also as a suggestion. But I will shut up right now. Are there any further questions?
Oh, well, I got a pretty simple.
Go ahead, Jesus.
Okay. On your first part of the talk, and you talk about you were using this mapper. Can you tell us a little bit of how you decide the parameters that are required to plot these graphs? Because it is my understanding that mapper requires a lot of parameters, like different clusterings, different projection functions.
Yeah. Well, this is not my work.
Okay.
But I understand the question because it's like a secret in most of these papers because they are not giving the details, the full details of the parameters on the mapper graph. I try to read in the paper, and one of the steps, maybe the dimension reduction, they use a patented algorithm that is patented by Google. So you don't have, well, if you buy the license, you can try to do try to replicate the results. But the information is not very clear in the paper. So you need to ask for the authors, this kind of information.
Okay. Okay. Thank you, Juan Carlos.
It's top secret science. Okay. Let's just move on. Any further questions? Okay. Well, in this case, Juan Carlos, thank you so much again. Let's thank the speaker. So I also do the virtual clapping. And well, I guess that's all.